With Maini, we are here to, let's say, chair this mini symposium that is entitled HBV Cure Promises and Challenges of Immunotherapies. So first, let me really thank the international coalition. Sorry. Yesterday night was a tough night for me. The International Coalition for Elimination of Hepatitis B virus to really give us the task to organize this mini symposium. And the mini symposium is organized in a very simple way, and again is promises and challenges. We will start with the promises, and basically we have three very well-known speakers that will touch three different, let's say, promises about immunotherapy. Vaccine therapy, and then we have an antibody-based therapy, and then a sort of, let's say, new approach using TCR-like antibodies. But let's start immediately with Ulrike Protzer. I don't even think she needs an introduction, but I will read a few things about her. She's basically the director of the Institute of Virology of Technical University of Munich, and clearly she has been working on the hepatitis B immunology and virology for many, many years. I think you know very well all her fantastic work. Thank you very much, Ulrike, to come to speak here.
Yeah, thank you very much, and also thanks to ICE-HBV and the Hepatitis B Foundation for the invitation to come here. So, but let me just briefly remind you how potentially Hep B can be cured. I named here all the drugs which we do have available, which is nucleoside analogs, capsid assembly modifiers in development. We have interferons, we have drugs which are supposed to induce interferon, we have siRNAs which are tested. These guys here, they target a relatively late step in the virus life cycle, so they would prevent that new viruses are formed, but they do not affect the CCC DNA that has been established. And similarly, interferons and also siRNA hardly do that. They mainly act on the mRNA, interference may have a little effect on CCC DNA, but we know that to cure it, we will not reach the doses clinically which we would need. And there is entry blockers which prevent the infection of new hepatocytes, but if your hepatocytes are already infected, if your liver is already infected, they also do not have any convincing effect so far. So what can we do here? Basically, you can think about, well, I simply kill all infected hepatocytes. You may have to be cautious because the liver may be affected before, and you should make sure that it's not too many cells which you kill. But we know also that in a healthy liver, you can remove 40% of the liver before you run into problems. We know that from split liver transplants. So I think there is a fair chance to activate T-cell responses without harming or without any safety aspects. That, if you kill infected hepatocytes, will induce cell division. And from a lot of studies from different groups, we know that cell division helps us to get rid of CCC DNA because you simply dilute it out. And then there's the degradation of nuclear CCC DNA by cytokines, which can be, for example, secreted by T cells or interferons or drugs which induce interference. And when you think about that, what could do all of the three things together, then you end up that a therapeutic vaccine is probably a very good idea if it activates T cells. And if you look at the disease and you see that there is cases which can control the virus, we would call it immune control, maybe instead of cure, and then there is cases which are immune tolerant, then those who control the disease, they have a strong antibody and a quite good effector T cell response. That correlates with self-limiting infection. while the others who are chronic carriers of the virus, they do not have neutralizing antibodies which we can detect. And T cells are really scarce. So there's almost no T cells. And what the therapeutic vaccine does, it tries to convert the immune tolerant state into an immune control state. So very different from a prophylactic vaccine. It really tries to modulate your immune system. The question always is, do therapeutic vaccines work? And I would say, so far, we don't know, because so far there's very few successful examples. However, hepatitis B is a perfect example. There is the possibility that a chronic infection is spontaneously cured. That's very rare, that's below 1%, but it occurs. So if we hit the right buttons, of the immune system, we should be able to make it. However, so far it did not really work, but people believe it will work. And that's why a lot of companies and institutions invest into developing therapeutic vaccines. And that's from the Hep B Foundation Drug Watch site, which I can really recommend all the currently ongoing trials on the left hand in the clinics and on the right hand still in a preclinical stage. Now the big question is, what type of an immunity should a therapeutic vaccine induce? Obviously the one we need to cure HBV, but what is it? Is it antibodies which neutralize the virus, prevent further spread, and may activate NK cells? I would say they are helpful, but probably not sufficient. Is it CD8 T cells, cytotoxic cells that kill infected cells in the liver, infected hepatocytes, or release cytokines? Certainly we need them, but maybe they're also not sufficient. Is it CD4 helper cells? They help T cells, but also B cells. They also secrete cytokines even more than the CD8 T cells do, but they can also be regulatory cells. And I'm quite convinced that we definitely need to activate CD4 T cells, because without CD4 T cell help, all the remaining arms will not come up. And last not least, it's B cells. And when we listened to Mala's talk yesterday, it might be very important to also allow B cell activation. And the question now is, how can a vaccine activate these different arms of the immune system, which somehow play together? And one of the key things is to activate the right antigen-presenting cells. If you do antigen presentation in the liver, that will not be sufficient. Otherwise, our immune system would kick out the virus, because there is high amounts of antigen circulating, but you need to activate different antigen-presenting cells. And the next question we have to ask ourselves, which tools do we have for therapeutic vaccination? And here in that slide, I try to basically sum up all the different tools that are available for vaccine development. And you know quite a lot of them from SARS-CoV or from COVID-19 vaccinations. And the ones we are using right now in clinical trials is, on the one hand, proteins, which, and that's lucky, in the case of HBV, form virus-like particles. So you can express S protein, and they would form HBs antigenous particles, and you can express core protein, it would form capsid-like structures, so core particles. And we know that these particles are much more immunogenic than the proteins per se. Then there is recombinant viral vectors, which are used, very similar to the AstraZeneca vaccine, which has been used for COVID-19. They have developed a chimp adeno vaccine expressing HBV antigens and use that in clinical trials, but also other companies are doing that using either Ad5 or Ad26 or even MVA. And the last thing, which is currently in clinical trials, is DNA. That is plasmid DNA, which has not been successfully used to develop a vaccine for humans so far, while the other approaches have been used, at least for prophylactic vaccines. But that's also an interesting approach to look at. The question, I think, which is around in the field is, will a single vaccine component be able to really break immune tolerance? And when you look at what is in the pipeline, only 3 out of 20 vaccine candidates are multi-component. 17 are single component. For an obvious reason, it's much easier to develop those and it's much easier to produce those. However, the big question is, will a single vaccine or a single component vaccine be able to do it? The next question is, will a single antigen be sufficient? Or do we not need to activate immunity against different antigens of HBV, which I'm quite convinced of? And the last question, which is also very important and which is also not fulfilled by a number of vaccines in development, is that they cover different HBV genotypes. We all know that in the US, genotype A is prevalent, but not in the rest of the world. There it's rather genotype D or B or C. And even E is a very important one in Africa. So one should really look at that a vaccine which works also covers those genotypes. Now let me address the question, is a single component sufficient? And that's just an example from our own work. And what we did is we generated an MVA vector expressing different HBV antigens, and we used that for vaccination, and we got very little response to HBV. A very nice pox virus response, so no monkeypox in this mice anymore, but that's not what we wanted. Then we used protein, particular protein, and core to vaccinate. And looking at T-cell responses, there is also not a lot. But when we combine the two, so protein and an MVA and a heterologous prime boost scheme, that works very well. And that induces nice T-cell responses and nice antibody response. And I think that observation has also been confirmed now in COVID-19 vaccine studies where heterologous vaccinations, like for example the British are doing right now, using an adenovector first and then an mRNA, that's a very potent approach. We, in our group, came up with a scheme we call TheraVac-B, and that's a heterologous prime boost scheme. So what are we doing? We basically are preparing a trial at the moment, and the clinical trial application is about to be handed in, and hopefully in January we can start to vaccinate the first healthy volunteers, and then right after that the first patients. And what, based on our data, we did is that we said, okay, we'll start with antiviral therapy, nucleoside analog therapy, because that reduces the inflammation in the liver, so it reduces the risk. But also, if you take that for a while, it restricts the number of your positive cells to hopefully the minimum. Those patients we then vaccinate with a particulate antigen, so S and core protein, recombinantly produced and adjuvanted with a suitable adjuvant to induce a balanced T-cell response. And this will be able to induce a B-cell response, so induce antibody responses and also activate B-cells outside of the liver in the lymph nodes. The antibodies are nice because they will prevent that the virus spreads further in the liver, but maybe also the activation of the B cells is an important component. And I think most importantly, it activates CD4 T cells. And protein, that's what all the old immunologists tell you, is just the best to activate CD4 T cells. mRNA also works, but protein works very well in that respect. And all our data point to the fact that this priming of CD4 T cells is really important. And once we've primed the CD4 T cells, we come with an MVA-based vector boost. And we chose MVA because it's just a very strong T cell boosting vector. MVA is a pox virus vector, modified not to replicate anymore in humans, has been tested in the 17th in more than 200,000 individuals and is regarded a quite safe vector. That's why we chose it. And we designed it in a way, or the whole vaccine in a way, that it activates immunity against genotypes B, C, as well as against genotypes A and D. And because genotype E is sort of a combination of the two, it also covers that. So if it would be successful, we would be able to apply it all over the world. Just an example how it works, and here we generated in a preclinical model of HBV infection and E-negative HBV infection, because that's mainly the patients everybody sees. We generated mice which replicate the virus, which have S antigen at very comparable levels. Obviously, the E- mice do not express E, but the wild-type mice do. And over time, the expression of those antigens and the viral replication in the liver is very, very constant. And we know that those animals develop an immune tolerance, and we asked the question, would our therapeutic B approach be able to break that immune tolerance? And Anna Kosinska found that if she vaccinates those two different mouse models, they develop anti-S antibodies. They clear S antigen to non-detectable levels, and those who had E antigen before also clear E antigen, and they clear all the core positive hepatocytes from their livers. That's the nice thing in mouse models, you can look into the livers. With relatively little side effects or inflammation in the liver, we do see a transient elevation of ALT. However, that's really transient over 2 weeks, and the peak we observed was around 200. And the pathologist told us, well, that's a mild to moderate inflammation, which you see, and that hopefully would be tolerated also in patients. Now, I told you we wanted to know what is really the important part of the vaccine. And to address that question, we did an antibody depletion of different T-cell subpopulations. And we depleted CD4 T-cells, and the results are shown here in the dots with a red filling. And we depleted CD8 T-cells, and that's the ones with the blue filling. And then we vaccinated the mice with a full scheme, so 2 times a protein, and then 1 time the MVA. And I think it's very obvious that if you deplete CD4 T cells, the vaccine does not have any effect. While if you do use a control antibody, it is fully effective. And if you deplete CD8 T cells, it did not have a lot of effect during the priming phase. And the effect, obviously, of depleting CD4 T cells is on the antibody arm, but it's also on the T cells. So if we deplete the CD4 T cells, then also the CD8 T cell response is much weaker. So the CD4 T cells are really needed for an efficient vaccination. And this E antigen scheme is a little more complicated because it shows that without any depletion, the vaccine is very effective. If we deplete CD8 T cells, it's only partially effective. But if we deplete CD4 T cells, it really loses its effect. And that's why we are so convinced that this priming step is a very, very important step here. And it's to activate the CD4 T cells. Now, so far we used protein, and now everybody talks about mRNA and also DNA. So we asked ourselves, already before the COVID vaccines came up, we started these experiments. Wouldn't be, for example, mRNA a very good substitute for protein? Would be easier to produce. And we could pack even more epitopes into the antigen. We could also prime for polymerase-specific T-cell responses, for example, which we cannot do with the protein approach at the moment. And we did so, and what we first did is we used DNA versus adjuvanted protein, and what you immediately see is that the antibody responses are much better if you use a protein, particularly the anti-S responses. However, even also when you look at T-cell responses, you see that the CD4 T-cell responses, particularly those for S, are better induced by the protein than they are by a DNA prime. And also CD8 T cell responses following that are stronger if we use the protein compared to the DNA. And this is results in mice, and we know that DNA vaccines work much better in mice than they do in humans. So we thought, okay, maybe that's not the best idea to use DNA because it was clearly inferior. But what's about RNA? If we now use RNA for priming and design that in an optimal fashion, then we found that antibody responses against core are very good using RNA. Against S, not as good as the protein vaccines are, but still at a significant level. But interestingly, looking at T cell responses, the RNA was able to induce quite a nice CD4 T cell response, and that's, I think, what it also does in COVID vaccination. But the CD8 T cell response was somehow poor compared to the protein-based approach. Still, I think it's an interesting approach to follow, and maybe by formulating the RNA in different ways, we already try to work with somebody who's professionally doing that, but maybe there is room for improvement. But for the moment, we decided our protein is superior to the other approaches, and we go on with the protein prime. Now, what we also observed, and I'd like to discuss that with you in the last few minutes, is that the starting titer of HBV antigens has a big role. And the nice thing about this preclinical model, the AAV-HBV model, is that you can infect those mice with different titers. And when you do so, then you end up with antigen expression and replication at different levels. And we vaccinated those mice who replicate HBV at different levels, and we found that the high replicator do not respond to the vaccine, or only in a minor fashion. The medium replicators, they respond but do not cure, whereas the low replicators, they completely clear the infection. And that corresponds very well with the induction of T cell responses. So looking at S and core-specific T cell responses, the high replicating, or in the high replicating mice, we were not able to induce significant T cell responses. There were some responses in the medium replicating mice, and there were very nice responses in the low replicating mice. So there's definitely a correlation with the replication level. So we asked ourselves, what can we do here? And we teamed up with, at that time, Alnylam to use their clinical candidate as siRNA and see what that does. And again, to just show you what the model is like, this is S antigen and this is E antigen. If you inject a control siRNA, nothing happens. Yeah, the mice replicate over 20 weeks. We had kept them up to a year. They replicate very persistently. When we now treat them with a very efficient siRNA, then we see a drop in S antigen of up to 3 log. That's 2 different candidates which we tested here, and a drop in E antigen of more than 1 log, almost 2 logs. However, as soon as your siRNA in fact fades and the last injection was here, the virus comes up again to exactly the same level as it has been before, indicating that even in such a model, which is not a lifelong carrier of HBV, but just a carrier for a few weeks, you are not able to break immune tolerance by just lowering the antigen levels. Now, when we vaccinate those mice, the interesting thing is that that's the ones which had only the siRNA in the dotted lines, and then the filled lines are those who are vaccinated in addition. And all those mice who received the siRNA and were vaccinated completely lost S antigen and completely lost E antigen expression. And interestingly, at the very end here, we had a little blob of E antigen coming up again, but then the T cells kicked in and it was gone. So that maintained. So combining an siRNA to lower antigen levels with a therapeutic vaccination may really be a very good idea. And this is just the antibody levels, which I think in this context are not so important. But what is interesting is, again, to look at the T cell responses, which finally are then the effectors that have to do the final job to really eliminate the virus. And you see, if we only use siRNA, there is nothing. But if we combine the siRNA with a therapeutic vaccine, we get very nice T cell responses, and that really reflects also the antiviral effect. And if you're not convinced yet, I'll show you the livers of those mice. And in the livers, you see the siRNA per se does not have any effect. That's the controls here. But if you combine with TherVac-B, then there's really almost not a single positive, core positive cell detectable anymore in all of the histologies we look at. And that's why we are convinced that really TherVac-B can cure HBV and control HBV in mice. We don't know in humans yet. But if there's high titers, you may have to pre-treat with an siRNA. So to summarize that, T cells are really scarce in HBV infection. And we are convinced that you need to activate and prime new T cells. Those who are there, if you just use a checkpoint inhibitor and take the break off those which are there, that will not be sufficient because there is almost nothing. But if you prime new T cells, then you have the option to really control the disease. It has been shown that therapeutic vaccine so far is safe, and at least in preclinical models has the potency to really cure. Activation of CD4 T cells is really crucial for an effective therapeutic vaccine, and in our hands that occurs during the priming step. Neutralizing antibodies help to limit the virus spread. Whether they also have a role in immune activation, whether the B cell activation plays a role, I cannot tell you yet. That's really something one needs to study. However, high antigen load over a longer time period will limit vaccine efficacy, and we need to think of selecting the patients with low viral load, which can be directly vaccinated, and those with higher viral load, where I'm convinced that we have to pre-treat them. So successful therapeutic vaccines will require a targeted design and a very elaborate selection of patients. Now the big question is, does the vaccine work in humans? And as I told you, we're going to bring that into humans in a consortium which is called TherVac-B, and that's the people who contributed to all the preclinical data. That's the project management team, which is really important, the immune monitoring team, and all our clinical collaborators, which I'm very grateful to, because it's a huge effort to do that. And we run a Be Aware campaign in parallel to our study, and try to really make a difference here. And with that, I'm at the end. Thank you for your attention, and thank the teams for doing all what they do, because translation is really a team effort.
Thank you very much, Ulla. There are many questions. I have also one question later, but please, Luca first.
Two quick questions. First question about the last immunohistochemistry, Ulla. My problem is that you show clearance of core from the nuclei, and in our models, in order to get rid of core from the nuclei, the only way is either stop expression for a long, long time, or force hepatocellular turnover. And the idea of forcing hepatocellular turnover in your model doesn't fit with the fact that there are not that many ALTs. So how do you explain the disappearance of core from the nuclei?
Obviously, the transgenic mice are a little different model than the AAV-HB mice. We started off with developing everything in the transgenic mice, and it has the same efficacy. However, because the transgene remains, your virus always will come back. That's why we switched the model and went to a model which is immune tolerant, but has the opportunity to clear the infection. And why the transgenic mice do have more core protein in their nucleus than we do have, I don't know, but definitely our mice also have quite a bit of core protein in the nucleus.
So basically, I don't get, what is the mechanism whereby in your mice you can clear core from the nuclei?
Potentially there's different mechanisms. So first of all, it could be cell division, where it gets out of the nucleus, because you have killing, right? If you kill the infected cells, then
Sure, no.
it's gone, and that's possible in our model, which is just not possible in the transgenic mice, because it will come back from the
low-level
transgenic.
killing for a long, long time. You don't pick it on the ALT, but then eventually you turn over most of the liver. The second question, very quick, is what really matters is how many hepatocytes are indeed infected at the end of the day. And so it would be wonderful, and S antigen is a lousy marker for that too in humans, and it would be wonderful to biopsy the patients before going in the trial and see how many core and S positive hepatocytes are there, and stratify then the results based on that.
Yeah, it's definitely a very good point, and really a word of caution using S loss in mice, because in our hands, the black 6 mice in particular, relatively easy to develop anti-S antibodies and clear away the S. You always have to look at the liver biopsy or at the E antigen. However, the E antigen is not available in most of our patients. That's the problem, because they're already E
the mice,
negative.
the relationship between S antigen and the number of hepatocytes.
That's why we rely on S. I don't see another way at the moment.
So I have two quick questions. You said that if you take out 40% of hepatocytes in split liver transplantation, then it's safe. I wonder whether in this setting we could even take out more, because you leave the stromal scaffold of the liver in place, and the regeneration obviously might be much more effective, because there will certainly be patients where more than 40% are infected and a target for the vaccine. And the second question is, as you obviously with an siRNA knock down also cell-expressed antigens or T-cell epitopes, whether you think there could be sort of a sweet window where you should end up with knocking down antigens with siRNA, which might be the most effective one.
Yeah. So to the first question, definitely I would not start with a patient who has more than 40% of its hepatocytes infected. In chronic infection, after a long-term nuke treatment, those patients are rare, but definitely you have to watch out, and we will be careful including patients with very high S antigen levels for exactly that reason. Because in a clinical trial, you're just not able to do liver biopsies. You will not be able to recruit. And the second question, the siRNA brings down the virus, the antigen load to a certain level. I don't know whether extremely high S antigen levels, which we are not able to reach in our mice, we can only go up to like S levels of 20,000, 30,000, that's it. So, and there's patients who have higher levels. Whether that would also, whether the siRNA would also be sufficient, I don't know.
Adam?
I'll try to be short. So, I mean, you've done a fantastic job looking at how these components go together to maximize immunogenicity, and it's super exciting that you're getting into the human trials. But I think you know that one of the problems in patients is that we get good vaccine-induced immunity that may not impact on the virus-infected cells because of genotype mismatch or vaccine. And I mean, the vaccine-induced responses may be good to boost that helper response, but I was wondering in your model, do you think you could test this? Because it's never really been measured using different genotypes in the AAV model to see how heterologous challenge with your vaccine actually relates to reduction of virus.
Yeah, it's a good point. We're about to do that. We've generated the AAVs with different genotypes to just mimic that situation. And our vaccine covers all the different genotypes in the protein part and also in the MVA part, which I think is very important.
So apologies, a somewhat naive question, but coming with a background in AAV-mediated gene therapy, this just strikes me as very much like transgene-specific responses sort of generically that we see. So I'm wondering what controls are done in terms of using other transgenes or otherwise just to kind of demonstrate that this is a specific effect toward HBV rather than if we'd had done this model with a GFP sort of transgene or something. And how do we know that this is fundamentally reflecting all the difficulties that cccDNA and HBV really cause in this infection?
Mm-hmm. That's a very good question. So what we've done, we've used control vaccines in all our experiments. So an MV which does not express any antigen and just a non-related protein, and then we do not see the effect. The other way around, using a model which does not replicate HBV, we haven't really done so far. We always use the HBV transgenics or the different AAV-HBV models.
Last question there.
Based
George?
on what of your data or experiments are you convinced that you need to prime naive T cells? And if that is really the case, what makes you hopeful that this is possible in patients after 20 or 30 years of infection?
I would not see a reason why it's not possible to prime those T cells. The big question will be, do they survive?
Do we know that we have a bunch of naive T cells after 20 or 30 years of infection?
I think so. Otherwise, they could not respond to other infections, right? And there is no indication that there is any immune deficiency against other infections.
No, but it needs to be obviously an HPV-specific naive T cells, and so those see the antigen for 20, 30 years, and you come in with a vaccine, and then you think the vaccine will do what the infection has not done over such a long time. And I'm just wondering whether there's any data to support that this is really feasible.
I think it's a very good question. Probably you as immunologists would have to address that. My answer would be there is a certain percentage of people who can cure HBV infection even after years of chronicity. And spontaneously, that means somehow they do it.
Right, but in those cases, the question is, it's not a reactivation of what they have, maybe.
Yeah. George, sorry, but very nice discussion, but we have also some time.
Just one follow-up question. I agree with Georg, and I think it's very important to select the population of patients you want to cure. So in an adult patient of age 40, 50 years, infected since birth, so for decades, all of the T cells that have an HBV-specific T cell receptor, which are there at birth, they have been primed, they have been deleted, they have been exhausted. And there's no thymus anymore to generate new T cells. So I think that's the underlying concern. If you take a young patient population, early 20s, I think the chances may be much higher because the thymus still generates new naive T cells.
Yeah, I think it's a very good point. Maybe we should discuss that over coffee.
Yes, good. Thank you very much. Okay, last very short.
Just one quick comment. There is an observational study where we do see e-antigen seroconversion in patients with chronic hepatitis B infection, and this has been tied to reactivation of T cells. So this is actually published. So I do think that although we have a hard time detecting those cells, I think they're there.
Yeah.
Okay, thank you very much, and thanks, Ulla, for a fantastic talk. Let's move on. So the next speaker is David Corti. He's the Senior Vice President at Vir Biotechnology. He's done fantastic work over a large number of years now on isolating, developing monoclonal antibody therapies for a vast range of different viral infections. You probably know his exciting work in COVID. But today, he's going to tell us about developments on antibody therapy for hepatitis B. Thank you so much, David.
Thank you, Mala.